Hello, my name is Claire Wu, and today I'm going to give a presentation about IgG-based anti-gp120 targeting bispecific T-cell engager antibody construction. HIV affects more than 38 million people worldwide as of 2019, and over 25 million of these people have access to antiretroviral therapy. HIV is a chronic condition that requires people to take medication daily for life. The reason for this is because antiretroviral therapies focus more on containment of the virus. So why is HIV-1 a chronic condition? HIV-1 is able to infect CD4 positive T cells using their gp120 envelope protein to enter the cell. The virus is then able to integrate their viral genome into resting CD4 positive T cells that then remain in the secondary lymph nodes until they are activated, and then the virus is able to hijack the cell's machinery and replicate. The use of antibodies in treatment of diseases and disorders has exploded the field of immunotherapy. Research on HIV has turned towards the search for neutralizing antibodies that are able to bind to multiple strains of HIV. Researchers are now able to take parts of the antibody protein and combine them to form new proteins that have different functions and specificities. It is therefore important to understand the structure of the antibody. An antibody is a homodimeric protein containing two light chains and two heavy chains associated together by disulfide bonds. The variable region is able to bind antigens, while the Fc region is able to bind receptors on effector cells. Second-generation antibody technologies called bispecific T-cell engagers, also called BiTs, are able to take these variable regions on antibodies and combine them to form single-chain variable fragments. Research has shown that it is possible to take the variable fragments of the heavy and light chain from HIV-1 neutralizing antibodies that target gp120 and combine them with a single-chain variable fragment of an anti-CD3 protein. An example of one of these BiTs is shown on the right. VRCO1 or B12 are gp120 neutralizing antibodies that are able to effectively bind to multiple strains of HIV-1. As you can see, these BiTs are a continuous polypeptide chain, as they are able to put both the light chain and heavy chain variable fragments together into one protein. Promising results in vitro data shows that it is possible to use these BiTs to target CD3 positive effector cells to activate and kill HIV-1 infected cells. However, there are limitations with the current approaches, as BiTs have a short half-life and cytotoxicity issues due to off-target binding. Our goal is to create an IgG-based antibody that is able to target these latently infected CD4 positive T cells. This antibody will maintain the IgG structure so that it will have a longer half-life and be more stable. Due to the heterodimeric nature of our protein, we will introduce knob-into-hole technology to the Fc portion of the antibody to help maintain the heterodimeric nature. As seen on the right, one of the heavy chains will contain a knob-like structure, another heavy chain will contain a hole-like structure. This will increase the likelihood that the heterodimeric antibody will form as two knobs or two holes associating together will not have a strong association as compared to a knob and a hole heavy chain associated together. So, our protein of interest should be able to bind to gp120 through the anti-CD3 single-chain variable fragment attached to one of the heavy chains. Our antibody of interest should also be able to activate latently infected CD4 positive T cells and activate CD8 positive T cells through the CD3 receptor. Our antibody will have two different mechanisms of action based on where the anti-CD3 protein binds. The first mechanism of action we will talk about is a cis mechanism. Our antibody will bind to HIV-infected cells, producing low titers of viral particles using the anti-gp120 variable regions from a neutralizing antibody. In our case, we have chosen 3BNC117 for its efficacy in binding multiple strains of HIV-1. After the antibody associates with the gp120 envelope protein, the anti-CD3 will bind to the CD3 receptor on the same infected cell and activate the infected CD4 positive T cell. This will cause even more gp120 to be produced and allow for more antibodies to bind to the infected cell. The trans mechanism of action is how our antibody is able to mediate the target cell killing. After the cis mechanism of action, infected CD4 positive T cells will have more gp120 expression on their plasma membranes. Our antibody will be able to bind to these cells more effectively. This will increase the chances of the anti-CD3 to bind to the CD3 receptor of a CD8 positive T cell. Binding to the CD3 receptor of a CD8 positive T cell will allow the T cell to become activated and mediate lysis of the target cell. Our objectives for this study can be divided into two phases: synthesis of the antibody and biological experimentation. Our first step is to synthesize a control antibody containing no anti-CD3 single-chain variable fragment and our antibody of interest with anti-CD3 single-chain variable fragment. Synthesizing the antibody DNA plasmid constructs will include the generation of 3 heavy chains and 1 light chain. We will then need to Sanger sequence our DNA plasmids to confirm the constructs are correct. After confirming our sequences, we will transfect our plasmid constructs into cells to produce our antibody. An enzyme-linked immunoassay is then used to measure our antibody concentration, and we also need to confirm our characterization of our antibody. Afterwards, we can then begin testing the efficacy of the antibody to kill infected cells. The methods used to conduct our objectives include molecular cloning using bacteria, DNA digests and ligations, plasmid mini preparations, as well as gel extractions. For harder constructs that were not able to be synthesized from cloning, PCR was used to amplify constructs before molecular cloning. Sanger sequencing was then used to confirm plasmid constructs. Transfection using polymer-based reagents were used to affect 293 T cells to produce our antibodies. Sandwich ELISA was then used to quantify our antibodies by using goat anti-human Ig for the primary antibody and goat anti-human IgFc HRP for the secondary antibody. From our results, we were able to sequence confirm all constructed heavy chain and light chain plasmids. From our sandwich ELISA, we were able to measure our antibody concentration for our control to be around half of a microgram per milliliter and 1 microgram per milliliter for our antibody of interest. Based off of the results, it would be hard to say if our antibody of interest was the one that we made. We will have to characterize the antibody to make sure our antibody has the 3BNC117 variable regions, the anti-CD3 single-chain variable fragment, as well as the heterodimeric nature. A Western blot or reducing SDS page will be the next step in understanding our antibody. However, from Sanger sequencing and successful production of antibody from the transfected 293 T cells suggests that the antibody was successfully generated, as fully assembled antibodies can only be secreted out of the cell, else the partially assembled or non-assembled antibodies are degraded within the cell. Thank you.